Okay, so good morning, and many thanks for inviting me here. So I'm always very happy to come back to Paris. Like many of the other groups that are speaking in this meeting, my group is studying host microbiome interactions. We regard the microbiome, if you may, as a signaling hub that integrates into it signals coming from the host genetics, the host immune system, but many other signals that come from the environment. You can see here some examples of these signals. The microbiome integrates all of these signals, processes them, and transmits them to the host. And we and others believe that these interactions are critically important for our homeostasis, and that breach of these communication channels between the microbiome and the host may lead to susceptibility to common multifactorial disease. Like many others, my group has tried to study these host microbiome interactions in the last few years. We've focused on developing pipelines that are aimed to use next-gen sequencing in order to identify from within these huge mixed ecosystem the driver bacteria that may actively participate in transmitting disease susceptibility. We've explored a lot of mechanisms related to innate immune microbiome interactions, and in the last 2 years we've mainly focused on trying to understand how nutrition affects the microbiome and how this nutrition microbiome axis affects metabolic homeostasis or the risk to develop metabolic disease. But today I'm going to tell you a story that is not included in this slide, but is part of our nutrition microbiome metabolism studies, which relates to the microbiome and circadian rhythmicity, as Brett mentioned before. And I will talk about some published stories that we published in the last couple of years, but also about some unpublished data that is maturing into a new story. And as many of you know, circadian rhythmicity and the circadian rhythmicity field has been blooming in the last 20 years with a series of discoveries that basically tell us that almost any function, any physiological function in the mammalian body is influenced in one way or another by the rotating exposure to dark and light. This field has managed to discover that we have a series of circadian clocks. The uppest one is the central nervous system central clock, which is regulated by light and by darkness, and transmits through a series of neuronal and hormonal stimuli signals to the periphery. But in recent years, it became apparent that, in fact, we have millions of peripheral clocks within our body. In fact, every organ in our body and every cell in every organ in our body features an independent clock. So we have, in any mammalian body, in the human body, for example, millions and millions of different clocks that in a very miraculous way are fully coordinated with each other and perform functions that are done in different quantities in different times of the day. And one more exciting development in this field came a few years ago when the molecular biochemical core clock machinery was deciphered, and this core clock exists within each one of our cells, and it consists of a series of transcriptional and post-transcriptional events of co-activation and co-repressors that basically change in different times of the day. So this is the clock, and I just want to mention to you this one transcription factor called period. Actually, there are two of them, period 1 and period 2, which are critically important and central in this core clock machinery, and we'll come back to them a little later in my talk. And one last functional importance to this circadian story is the fact that in the medical literature, there is extensive evidence in the last 70 years that people, post-industrial revolution, populations of people that feature disturbances in circadian activity, such as shift workers, soldiers, people featuring severe jet lag, like myself, are actually dramatically at risk for the development of obesity, type 2 diabetes, and other features of the metabolic syndrome. And we have no idea what the missing link is between environmental circadian disturbances and the risk to develop these diseases. Now, since we in my lab regard the microbiome, if you may, as a neglected organ, we started by a very naive question, which is, does the microbiome features like any other organ or cell in our body a circadian activity? So Christoph Theis, in collaboration with Maayan Levy and Tal Korem, took upon this very difficult challenge, and to make a very long story short, they performed a series of characterization experiments of the gut microbiome at different times of the day in repetitive days, and found that the gut microbiome features a very significant diurnal activity in many different levels. You can see here an example done at the compositional level. This is 16S ribosomal DNA analysis of gut microbiome in wild-type mice done at different times of the day. This kind of readout is something we get by applying a very stringent statistical tool called JTK cycle, which basically tells us if a community behavior is cycling or non-cycling, while being very stringent in avoiding false discovery rate, which is the biggest enemy of these circadian papers. And anything, any dot, any bacterial taxa below this dotted line is statistically cycling or circadian in its behavior, and you can see that around 15 to 20% of all bacterial taxa in the gut microbiome feature a very reproducible diurnal behavior. You can see here an example of one of them. But this is just the compositional level. Now when we go into the functional level, when we perform shotgun metagenomic sequencing, in which, unlike 16S, we sequence all the genes of all the bacteria in every configuration at different times of the day, you can see that when we assemble gene content and pathway abundance, we equally see circadian activity in a large minority of functions. You can see here an example of some microbial functions that go up, down at night-day cycles in repetitive days. And this was not all. Even if we look at more complex, higher microbial functions, which take into them different microbial pathways, such as flagellar assembly, you can see that these complex functions can show circadian activity. So all of these pathways go up or down at the same time of the day to bring along this higher microbial function. So this told us something very, very interesting about mice, and we wanted to see whether this is this cycling diurnal behavior is also true in humans, and you may imagine that doing this kind of experiment in humans is much more technically challenging, but we were lucky to find some good volunteers who gave us their stuff every few hours for many different days, and you can see that when we sequence them both in the compositional and the shotgun levels, you can see, and this is an example of one of the participants, that even in the human, the gut microbiome features a diurnal activity with taxonomic groups going up, down, in this case in 5 consecutive days, and likewise the KEGG functions in these configurations. Now this was very surprising to us, because if you think about it, the gut microbiome lives completely in the dark, so how do they know that it's light and dark, and how do they behave accordingly? And to answer this question, we returned to the core circadian clock that I've mentioned before, and we've utilized mice that are deficient in the period 1 and 2 proteins. So these mice have been shown before, way before us, to completely lack a circadian activity of the host. So they have absolutely no host circadian activity, and we can use these mice in order to see whether the lack of a host circadian clock affects the microbial circadian clock and regulates it. And the answer was a clear yes. You can see, for example, in this experiment, when we compare the gut microbiome, composition which we sequenced at different times of the day, to that of pair 1-2 knockout mice, you can see, as I've shown you before, that the function of the gut microbiome in wild-type settings is highly rhythmic, going up, down in different consecutive day-night cycles, while that of the period 1-2 knockout mice becomes completely random, and a complete loss of the microbial rhythmicity is featured, which tells us that somehow the host molecular machinery is controlling the microbial molecular machinery. Now, to go further, these mice have been described, again, in the circadian literature way before us, to feature a secondary severe deficiency in their cyclic eating behavior. So these mice do not eat like regular mice only at night and don't eat during the day. They eat like a 24-hour buffet. They don't eat more than regular mice, but they lose their eating rhythmic behavior. So we ask ourselves whether this pattern is actually due to the fact that these mice lose their eating behavior. And to answer this question, we performed a series of force-feeding experiments in wild-type mice, in which we fed them only during the day, or only during the night, and repetitively sequenced their gut microbiome. And you can see that we can completely control the phase of the cycling microbiome by controlling the phase of the food intake by these mice, telling us that probably it is the feeding rhythmicity that is controlling the microbial behavior. And in fact, if we take these pair 1-2 knockout mice and we force feed them only during the day or only during the night, we completely restore their normal microbial circadian rhythmicity. Now, this had a lot of functional importance, and in order to prove this, we used both mice and humans. In mice, we induced this very robust model of jet lag in which we moved mice across light and dark cycles. If this would be extrapolated to humans, this would be like taking a human and flying him over from Paris to Beijing and back every 3 days for 7 consecutive times, for 21 days. So this is quite a severe jet lag. And in humans, what I've done is a set of experiments which actually made me the most popular young PI at the Weizmann Institute for 2 weeks, because I took a group of graduate students and I flew them all over the world, and I only asked them to collect their stools while they were jet lagged during the process. And we repeatedly sequenced both the mice and the human volunteers, and we've seen that during jet lag in both species, we saw a very significant disturbance in the cycling behavior of the microbiome. But to go into functionality, we performed a stool transfer experiment in which we transferred microbiomes from jet lagged or non-jet lagged mice, or from jet lagged or non-jet lagged humans into germ-free mice, and measured their ability or the rate of which they develop obesity and its complications in a high-fat diet model. And you can see both in the animal models and in humans that during jet lag, or the transfer of a jet lag microbiome induced obesity and glucose intolerance. This is in mice, this is in humans. In humans, you can see that when we do this with stools that was collected after the resolution of jet lag, we go back to baseline levels, at least implying that maybe a partial explanation to this striking association between circadian disturbances and the propensity to develop obesity and its complications may be driven by the cycling behavior of the microbiome. So this was published, but it also opened many interesting and potentially important unanswered questions. One of these questions, for example, was, we show that the microbiome oscillates, but how does it oscillate? So one could think that maybe bacteria die in different rates during different times of the day. Maybe they proliferate at different times, and there are many other potential explanations. This is very hard to, or difficult to answer, because most of the gut microbial components of the gut microbiome are very hard to culture. So doing classical experiments is very difficult. So a very talented graduate student, Tal Koren, took upon this challenge, and he actually came up with a very nice trick to take a static shot of next-gen sequencing and to extrapolate from this data set something about the proliferative behavior of each bacteria within a complex microbiome ecosystem. And the trick was basically dependent on the, or based on the fact that most microbes feature a single origin of replication, and the fact that the more proliferative a bacteria is, the more copies of its genomes it will make before it actually divides, which allows us to actually count the genes that are closer to the origin of replication and to divide it by the genes that are farther apart from the origin of replication. And this ratio, which we call peak to trough ratio, is actually very much accurately associated with the proliferative bacteria of a given bacteria. So this allows us, from a snapshot of a non-culturable, highly mixed microbial ecosystem, to tell what is the rate on a cell intrinsic basis of each of the components of this ecosystem. And this allows us to now study many different disease models and try to identify the driver bacteria, which we assume are more proliferative at a given context from this huge mixture of bacteria. But in our context, the circadian story, it allows us to show that, in fact, this hourly shift in bacterial composition is driven, at least partially, by differences in microbial proliferation. And you can see here examples of 6 bacteria from a shotgun sequencing experiment, and they all feature these very nice proliferative peak-to-trough ratio differences. An equally important and unanswered question that stemmed from our studies goes back to the host. So I've shown you, examples of how robust the cycling behavior of the gut microbiome is. But if you think about it, we have now 2 very stringent, very robust, hardwired circadian systems. You have the host on the one side, and you have the gut microbiome on the other side. And the obvious question is, does the circadian rhythmicity of the microbes affect the host, and how does it do it? What is the mechanism by which the cycling microbial behavior is affecting the cycling behavior of the host? And this, in our previous studies, remained a big black box. So to really start and scrape the surface on this, we shifted our attention from the luminal microbiome, which is something that we and most of the researchers in the microbiome field are very comfortable sequencing because it's very accessible, to the layer of bacteria that are firmly attached to the epithelial cells at the gut, which we assume are, in fact, the bacteria which are most actively engaged and communicate with the host. So these are the bacteria that if the microbiome in its circadian behavior is doing something to the host, these are our main suspects. So we repeated this series of very laborious circadian experiments at different times of the day, trying to see whether this mucosally associated community of bacteria is also featuring diurnal behavior. We started by doing a series of scanning electron microscopy to look at these bacteria, and to our surprise, we saw that the number of attached bacteria to the lumen very significantly shifts in its number at different times of the day. We repeated this again and again, and it was always very true. We validated this surprising finding by quantifying the number of attached bacteria by qPCR, and you can see the exact same phenomenon, and by next-gen sequencing of this attached population of bacteria, which again show that by composition, bacteria are changing their composition and their relative numbers in different times of the day when looking at this population of attached microbes. We also sequenced these bacteria not just by 16S, but also by shotgun metagenomic sequencing to start looking at their genes, modules, and pathways. And like before, and even more strikingly than before, we saw a very significant number of microbial functions that are very circadian in their behavior, going up, down in different times of the day. Among them were very interesting functions related to motility, such as flagellar assembly, chemotaxis, and mucus degradation. I will not show you further data that we have on this, but at least one of the mechanisms that we think are involved here is differences in microbial motility and mucus degradation at different times of the day. And since we believe that one of the main features by which the microbiome, at least the gut microbiome, affects host function is by the secretion of hundreds and thousands of different metabolites, we went to the trouble of performing an around-the-clock metabolomic assay in which we quantified thousands of different metabolites at different times of the day. And like in the other levels of microbiome functionality, you can see that many hundreds of metabolites feature a very strong circadian activity going up and down in different times of the day. You can see here a nice example of one such function which kind of bring together the genetic and metabolomic results, biotin biosynthesis. And you can see that there are two rate-limiting enzymes in this pathway in bacteria. You can see that both of these rate-limiting enzymes feature circadian activity in their levels in the shotgun metagenomic sequencing, and the resultant biotin is also highly circadian in its activity, and there are many other examples like this. Now, if this mucosally adherent microbial diurnal behavior is affecting the host, then we should now turn to the host and see what happens when this bacterial activity is disrupted. And in order to do this, we performed a series of experiments in mice in which we either use control mice or administered them wide-spectrum antibiotics to disrupt their gut microbiome. And we performed a series of transcriptome experiments, characterization experiments in the host gut using RNA-seq. Now, when we give broad-spectrum antibiotics, as expected, you can see that the microbial load is dramatically increased, but you can also see that this normal diurnal activity of the bugs that remain after this wide-spectrum antibiotic treatment is completely disrupted. You can see here in this JTK cycle readout that these are the bacteria which are normally shifting in control conditions, and when we give antibiotics, these are the remaining bacteria which lose their diurnal behavior. Now, when we look at the host and the host gut transcriptome, we were surprised to see that the fact that mice were exposed to antibiotics did not change the total number of cycling transcripts. So the total number was roughly the same. However, when we looked at the identity of this transcript, you can see that the transcript changed in their identity from control to antibiotic conditions with a group of transcripts that are present and are cycling in control conditions and are non-cycling once you give antibiotics, a group of transcripts that are cycling no matter what we do. So if you give control or if you check them in control conditions or in antibiotic treatment, nothing happens. But the most interesting is a group of transcripts that are normally non-oscillating and become oscillating upon antibiotic treatment. So the host assumes a cycling behavior of these genes following disruption of the microbiome. And you can see here the heat maps showing the different groups. And what was very interesting is that the core clock components, those components that I've shown you before, which run the clock, are completely oblivious. They don't care about the antibiotic treatment. They remain cycling, so the core clock becomes fully functional upon antibiotic treatment. But many genes are lost upon antibiotic treatment, and this is the interesting group with many functions that are gained and become to oscillate upon antibiotic treatment. And what's really striking for us was the fact that many of these functions are functions that we found to be normally oscillating in the gut microbiome itself. And once you get rid of the oscillation in the gut microbiome, they kind of appear to be oscillating in the host. So this suggests, and this is pure speculation, that perhaps some of these functions are normally buffered by the microbiome, and once you disrupt the microbiome, the host assumes these functions. But again, this is pure speculation at this point. Now, in order to dive a little deeper into the mechanisms by which the microbiome affects the host's circadian rhythmicity, we went and performed a series of epigenetic characterization experiments in the host, thinking that this would be a good suspect for changes that occur in an hour-scale resolution. And we looked at active promoters, active enhancers using ChIP-seq experiments in isolated gut epithelial cells. And just like I've shown you in the transcriptome assays, when, for example, we look at active promoter activity using K27 acetylation in H3K4 trimethylation in epithelial cells, you can see that many of the active promoters in the gut are normally cycling in their activity, and this is way above the background noise as is seen here in the control. And you can see the same exact phenomenon that I've shown you for the transcriptome before, that once you induce disruption of the microbiome by antibiotics, you all of a sudden see that some active promoters are lost in their circadian activity, some stay rhythmic, and some assume a cycling behavior which was not apparent before. And you can see here an example of a core clock component, NR1D1, which stays cyclic in different times of the day in consecutive days, and examples of the other two groups. The same is true for active enhancers, which assume upon antibiotic treatment a loss or gain of activity, and we've measured it in two different epigenome experiments, with a minority of them remaining cycling, and these are the core clock, actively core clock genes. And to put everything together, when we look at the cycling transcripts and the cycling active promoter and active enhancers in each of the two conditions, you can see that there is an enrichment of the cycling promoters with the same set, the same exact identical set of active transcript and active enhancers, implying very strongly that this active epigenetic cycling program that we uncovered is responsible for the identity of the cycling transcript at the different conditions with and without antibiotics. And finally, this was all done in the gut, but we know from the circadian literature that everything peripheral is very circadian. And we wanted to see whether we can, whether the cycling microbiome actually has an effect beyond the gut on other features of the host that are cycling. And we decided to focus on two such functions. One is the cycling behavior of serum metabolites and the cycling behavior of the liver, the hepatic transcriptome. Now we know that up to 50 to 80% of our peripheral blood metabolites are contributed by the gut microbiome. And we also know from previous studies by others that many of these metabolites are very cyclic in their behavior. And we repeated this by a metabolomic study in our hands. And you can see here an example looking at amino acids and polyamines showing that virtually all of them are very diurnal and rhythmic in their levels. You can see in the peripheral blood that they go down, up in different times of the day. And when we give antibiotics, these blood metabolites completely and dramatically lose their circadian behavior. You can see that this is the level, these are the cycling metabolites under normal conditions, and when we give broad-spectrum antibiotics, we completely lose this circadian behavior. Now you may ask, and actually our reviewers asked as well, whether this could be a result of the antibiotics themselves having a direct effect on all of these features, and the answer was answered by a very painful series of germ-free experiments, which we repeated all of these, and I can tell you that the results are practically identical, meaning that it is not the antibiotics, but the lack of microbiome or microbiome cycling behavior that is driving these effects. And when we looked at the hepatic transcriptome, just like the gut transcriptome, we could see that upon antibiotic treatment, we see the exact same unexpected behavior with many transcripts in the liver losing their cycling behavior, some retaining their cycling behavior, and a very large group obtaining a newly acquired cycling behavior upon the disruption of the gut microbiome. And just like I've shown you in the gut, when we divide them by heat maps into the three groups, you can see that the core clock components are completely maintained in their normal behaviors in the liver, no matter if you give or not give antibiotics, but you have a lot of microbial functions that are lost, and a lot of microbial functions in the, a lot of host functions that are lost, and a lot of host functions in the liver that become cycling upon disruption of the gut microbiome. And based on these results, we can start to draw a map or an atlas of hepatic functions under normal conditions and under conditions in which we disrupt the gut microbiome. And many of these functions now occur at the wrong time of the day, and there is an overwhelming amount of literature from the last 2 years from the circadian field suggesting that these time disturbances in normal function in the liver actually drive pathogenesis of differences in drug metabolism and susceptibility to liver disease. And these results at least suggest, and we're working on it right now, that perhaps some of these circadian disturbances may be mediated by the gut microbiome. And just to summarize what I've tried to show you, and this is a nice review published by the Paolo Sasson-Carsi group just a few months ago on our story and on many other follow-up stories which were published on this microbiome circadian rhythmicity. We are starting to think of gut homeostasis in a different way than we thought for many years. I at least usually thought of homeostasis as a static condition in which everything is stable and nothing changes. But what our and other people's results are suggesting is that we have this amazing tango between an hour-scale change in the gut microbial function and composition and the host activity, which are closely and bilaterally linked together. We call this shifting homeostasis. And this shifting homeostasis is affected by the central and peripheral circadian clock, but by what we eat, by when we eat what we eat, as I've shown you, and by, of course, the microbiome composition and function. And this tango, this shifting homeostasis, has profound effects on many functions of the host, which we're just trying to start to learn. And of course, this system has profound importance in many different aspects of how we see the microbiome in its interactions, but it also has very down-to-earth kind of technical implications, because if your postdoc does an experiment, takes the microbiome in the morning, but actually collects the host-related tissue in the evening, then he or she will see many differences that may be spurious and actually be a result of taking this shifting homeostasis at different time points of its existence. And I will finish by thanking the many people who have been doing this work, many people from my lab and from the lab of my good friend and colleague Eran Segal, who is a mathematician from the Weizmann Institute, are involved in these complex projects. The people who have led these two projects are Maayan, Christoph, and Tal. Actually, Maayan and Christoph from my lab have been working so hard on this project for the last 3 years that I just found out a couple of months ago that they became a couple. So, you know, it's another form of German-Israeli cooperation. I would like to thank all the funding agencies that are making this work possible, our clinical and other collaborators, and I think I'll stop here. Thanks.